now it's, I guess, time for the discussion. So feel free for the audience to communicate questions. I'll start off by querying the whole group further about the idea of reducing dimensionality, since that seems to be a common theme. Sui Wang yesterday talked about characterizing a cell state in terms of a state vector. I think the 2005 paper that he authored with Don Ingber in Physical Reviews Letters, not your usual, not a holding of the FDA library, I think. It was the very first use of this term, and they suggested that the vector would contain 2,000 to 3,000 elements to fully describe a cell transcriptome. But let me ask the group, do you think that number of elements is really necessary to convey most of the information? Because A, many of them are not relevant to the distinction between states, and B, some of the others could be collapsed, I think Ilana touched on this, into a simpler signal transduction pathway elements.
I could start with that. So we've been doing a lot of work in factorization approaches, and we and others have been starting to show that, you know, the dimensionality is really a function of the biology that you want to capture. And that it's not that there's one right or one wrong dimension. It's that what is the dimension that you're trying to uncover in the system? And so even back in bulk days, for example, we found that if you were using 2 dimensions in a system to represent it, you could separate, for example, tumor and normal. And then when you increase the number of dimensions, you were able to get from separating tumor and normal to separating out different tumor subtypes. And so it's really that there's a hierarchy of dimensions in biological systems, it seems like. And so it seems like you need to use a hierarchy of methods in terms of sort of multi-scale dimensionality to really uncover the system. And so it's probably low dimensional, but fundamentally multi-scale. And then it's also that different computational methodologies are also going to give you different dimensions or different features. And so it might be that you need to use different latent space methods depending on what you want to uncover. So I think that it's fundamentally yes, it's low dimensional, but which dimension are you trying to capture?
I think that answer suggests substantial job security for bioinformaticians. And will be cell type specific, or from the standpoint of an FDA person, product specific. Okay, another question that I think speaks to certain of the limitations of existing methods is the question of, does a 0 in a data matrix mean no expression or, quote, dropout, unquote, a technical issue related to sampling? Relatively few have actually tried to address this explicitly. I think the Fabian Theis lab has used an autoencoder to try and address that. I noticed an autoencoder in one of Paul's slides. That seems to me a good start, but I think a lot more attention would be helpful. What do you guys think?
So since you mentioned my presentation, I can tell you a bit how we deal with that. So our data, our pipeline allows you to do a lot of, to redo the experiments many times and to account for this kind of problems where we would miss potentially some data points. So we do have a way to somehow quality control when we see that we have points missing, and we even develop some ways to use our data set to correct if there is any issue, but still in a reasonable and coherent way to make sure that when we're missing a data point, I mean, either we can account for it, or we just we don't know that the experiment has not been working. So I guess our method is a bit particular in the sense this is not really like single-cell RNA-seq where we look at many dimensions at the same point. We're slightly lower dimension, so we can do that a bit more systematically. But certainly these are some very good questions that we need to address. And I will.
Picking up on that a little bit, I think it was Ilana who mentioned what amount to the use of semi-supervised methods. Sometimes, if we're looking for the presence or absence of an activated signal transduction pathway, we might, for sampling reasons, not capture all of the elements we're looking for, but maybe one or another of them might give us a clue. And this 1 or 0 thing strikes me as something of a logistic regression problem that in a neural network type approach could help decide whether a neuron is fired or not. Do you think there's any sense in pursuing that sort of idea?
I mean, I think it's definitely something that we could, start thinking about. And I think this idea of embedding architecture into systems to start understanding where things are occurring or where they aren't is going to be very important. And I think, even in factorization approaches, you're going to get different features if you encode different prior knowledge into the genes that you're starting to factorize. And so I think this sort of multi-scale nature is going to be critical to really uncovering what's going on in a system. And, it's always been a question in genomics as to where you flash your, where you sort of shine your flashlight in terms of understanding it. If you're focusing very deeply on a cell type, you're going to be able to uncover deeper knowledge. And I think that's something that's underappreciated in genomics, is that the analysis of data needs to be just as hypothesis driven as the experiments that are going in if you want to uncover something.
That makes perfect sense to me. Okay, now we've got some questions from the audience. This is from Anne to Paul. Does the projection space that works for visualization of correlations in the data provide any insight into the biologic process or network?
Oh, absolutely. I did not have time to talk about this, but definitely, like we see. So I briefly mentioned that there is something about the adaptive cytokine versus the adaptive versus innate cytokine, and we definitely see that the manifold where we project the data is related to the magnitude of one versus the other. And that's something we're actually exploring more right now, because we believe there are very, very interesting biological information on where this low dimension manifold is sitting, because it might vary a little bit between different antigens, so and different T-cell receptors and stuff like that. So this is something we're exploring. But yeah, it's a very good question. We're definitely moving in this direction.
Okay, another question for Paul from Kimberly. Can the immune velocity be used to phenotype the immune health of a patient? What is the effect of age, gender, pre-existing condition on immune velocity? We are still in the process of exploring all those questions. So we are not at the stage where we're going to look at actual patients and see how it's working. We are more this day we're looking at different T cells, different T cell receptors to see how this immune velocity is defined. It's actually a property of the antigen itself, so not necessarily of the T cells, but on the patient. But our hope is that our tool will allow to quantify those things in various contexts. So really, it's more like a tool to measure things, and then down the road we will try to see how we can apply that to different contexts. And how, a related question, how about using immune velocity as a functional test, for example, for immune system directed cellular cancer therapy.
Yeah, that's exactly what we're trying to do right now. And
Well,
this is really what we're trying to do.
Keep us in the FDA in that loop,
Okay.
please.
Great.
Okay, a question for Anne. A lot of developmental processes are by nature modular. All the way back from classical limb transplantation experiments and so forth. It seems that nature has anticipated the concept of unit operations as taught in chemical engineering seminars that get advertised in my mailbox all the time by millions of years. Do you think that the idea of sequential attractor states might help simplify process design?
Yes and no, because I think we should have more global picture, and is that you need to have like think sequentially, and at some point just like make a step back and don't forget what is before and what is after, because this is the problem of thinking everything as a single unit operation. Because if you just focus on, I want to optimize my process and I have this and this step to optimize, and you can optimize every single step, but you don't think back already on what will be the influence on your drug directly on, because like at my level, I optimize the condition for an organism. This is really great, but if I don't take into account what influence it will have on the molecule that I produce, and this influence, how it influences the efficacy on the drug, there is no point of optimizing, and potentially the optimality is not what we search. We may search suboptimal solution, but that might be optimal for the disease.
Okay, very good. Now a question from Krish. How do you think that machine learning and artificial intelligence black box modeling can be merged with systems biology? How should the communities talk and develop these tools? I guess that's for the whole group. Oh, no, it's for Anne, once again. Sorry.
There is a lot of people beginning to approach this problem of what we call the hybrid model to like specifically, like, for example, the metabolic models I'm using to describe the metabolism, it's one point, but they do not integrate the influence of process parameters and critical process parameters that are related to our process. Then if we wanted to integrate, for example, I will take a very simple example, the temperature and the influence of temperature on metabolism will not introduce, I don't know how many kinetic parameters to see how it can influence every single reaction. We'll need to try to kind of lump all that in a kind of single statistical effect on globally what you observe. And doing so, I think there is part, again, as we say, we could develop a very complex model to do that, but is it what we want? Do we need to do that? For understanding the process, it will be very interesting, but after to control and develop a process, it will not be necessary. Then is again, as we say, all the models are right, and but some are useful. It's,
Yes. Okay. And another question that I think has not been asked yet, also for you. Can the biological systems approach you delineated in the integrated graphic start with the patient? And although it is an N of 1, that individual represents many complex and interacting biological activities. Thus, what if we position the iterative regenerative medicine drug development process as starting from responders versus non-responders as the ultimate goal as a broadly successful suite of therapies?
This is a really cool question. It's actually like this idea of like having actually, when you think about a model, I think you should begin the model at the level of disease and take it with you along the process. And it's for that, then actually you could have models that are person specific, because if you begin with the disease and you know the influence of the genetic, age, sex, or whatever influence factor you want to take into account, you can actually make a lot of parallel aspects. And if you have all these influence factors that are connected, you could like really think of fast development of personalized medicine. And I think it's really one of the goals is to say you take a sample of your DNA and be, okay, you display this part of the disease, we know this influence, and we can like target what will be the most appropriate quality attributes of this molecule that need to be in it, such as you can really target most efficiently your disease. And after you have everything, as you know what you want to produce, you could like, and you know all the influence factor and their interaction, you could theoretically produce very fast a drug that will be completely focused on the expression of the disease in a specific patient.
Wow, that is indeed a fascinating question. Okay, I'd like to return again to the question of limitations of current methods. A lot of methods for looking for different cell types are based on clustering algorithms that treat all transcripts identically, when in fact the determination of one cell fate versus another may not depend on whether it's expressing aldolase, for example, but whether but much more importantly on whether BMP or Wnt signaling are activated. Classical algorithms for comparing protein sequences recognize that changing an aspartate for a glutamate is nowhere near as consequential as changing an aspartate for a phenylalanine. But relatively few clustering methods make any attempt at this. Does anyone have any ideas how to address that issue?
I think for part of it too, that's a big part of why we've gone to sort of these latent space representations and matrix factorization as opposed to clustering, is because it explicitly allows for gene reuse, right? So one of the big limitations that we think in a lot of clustering algorithms is that you're forcing either gene A to be in one class or another, whereas we all know through biological systems and systems level approaches that genes are reused through multiple processes. And so that's what we think is one big advantage of matrix factorization. Of course, the challenge is then, how do you know if it's real, or how do you know which cell type it's going to, which is why we've been thinking about the sort of transfer learning approach, where if you go from one cell type to another, can you benchmark this in data sets where you have a known ground truth of that cell type, so that you're relying not on a single gene, but rather gene signatures and how they're preserved across data sets and across cell types. You also brought up the sort of impact of scale and sort of function within a single transcript. And I think that's something that's largely ignored even with a lot of matrix factorization techniques, is people sort of throw in a lot of the data to these factorizations and hope that it works. Something our COGAPS approach has done that's a little bit different is to explicitly encode the uncertainty in each matrix element that you can have as a variable with it. And so you sort of scale for how much a transcript's expressed and try to downweigh it so that you don't just have the most highly expressed transcripts biasing it. And so we think this is where the semi-supervised learning approach is going to be really powerful. Of can you encode, different features into the system to try to understand, what are sort of the relevant factors in different systems, and not make it in a way that's just biased by the most highly expressed or most correlated genes.
Yeah, I think the limitation of dominance by things that are abundant rather than important, comparing, for example, transcripts for structural proteins versus receptors, transcription factors, and so forth, hasn't received enough attention. And there's a lot of residual convention, if you will, of practices like zero mean and unit variance, when those important genes often are regulated in a bistable manner how much sense does zero mean make in that context? Maybe you can comment on that and also connect that to the idea that multi-omic data, if it too is regulated in a bistable way, might be amenable to relatively simple scaling as ones and zeros.
Yeah. So we've thought about that in terms of, again, sort of weighing the system down by the uncertainty matrix. And then you can also use that to try to model regulation specifically, where you start thinking about, different modalities have different error terms, and you can include that in an integrated framework. It looked like Paul also had a comment that he wanted to bring up with this.
Yes, just a short comment, which is related to things we've been doing. And since I've mentioned it during my talk, I also think for all this question, being able to maybe follow a few genes, fewer number of genes, but with time, really the same experiment can be really, really useful because, if you just compare different experiments at different times, you have a lot of, noise. That you might not control. But if you can follow a trajectory over time, then you might see a very interesting geometric feature in the data that are very helpful. So for instance, you were mentioning bistability. Yesterday, we had a talk by Sui Wang, who were mentioning the criticality when you go to a monostable to a bistable phase. Those things will be, you could see them as you follow in time. And in my own talk, we're really following the same cells as a function of time. And this is how when you follow the entire trajectory that you can start seeing interesting features appearing. So, in development, I've been working a lot on modeling and study of developmental system. Time is really, really crucial.
Perfect. Fascinating. Well, that brings us to the end of our allotted time for the discussion. I'd like to thank everyone. I very much enjoyed everyone's presentations and the discussion, and I hope we can continue this later.